Hello everybody and very good day. I promised to talk about some training in microbiome analytics, and this is especially from the perspective of the cost action and machine learning for microbiome analytics, where we are involved with, and of course also related to some earlier training activities that my team has been running. So training in microbiome analysis is a versatile topic, as you have already, of course, heard many overviews on different aspects of this also today. Within this machine learning for microbiome action, we have aimed to make some overviews on the current challenges and topics in this area. But of course, as always, biological systems and ecosystems of microbes are very complex, and it is not an easy challenge to teach and train such multitude of techniques and concepts and practical skills that are needed in this kind of setting. So we can easily think that we teach algorithms and then it can be applied to some problems, but of course you have to see the full picture. And the training activities, of course, are part of teaching that full picture. I'm coming back to that. But if we look at some elements of microbiome data science, there is nothing strikingly different from other areas of bioinformatics or systems biology. We have data, typically lots of data, multi-omics, different measurements, functional and taxonomic measurements, for instance. And then we have the sets of methods that we use for data exploration and analysis and reporting. And often this cycle is, of course, iterative. So when we start to analyze data, we understand more and we may need to change our methods or upgrade our methods complement. So these elements, in principle, at high level, are clear. We already have many existing methods and techniques to analyze microbiome data developed during the past decade. And teaching of these methods is often based on teaching of similar methods in other areas of high throughput data science. But now in microbiome research, we realized that there are some specific properties of microbiome data that make this different from many other areas of bioinformatics. Of course, some of these are also common, but some of the topics that are specifically emphasized in microbiome studies are the effects of finite sampling. We always have, we more typically work with read counts, so we can always see just a random subset of the community from the modern measurement techniques. Then we have to take into account the different hierarchies in the data. For instance, we can consider hierarchies in the phylogenetic space. Different bacterial species or other microbial taxa, they have various relations. And this is important to consider. In the methodologies, we can also have hierarchies in the sample space if we study nested study designs, family hierarchies, and so forth. And then microbial data is compositional, and there is a vast literature on this topic as well. And not only that, but then we have a variety of study types ranging from data pre-processing issues to case control studies, interventions, and large-scale population level studies. We have aspects of temporal variation in time series, spatial variation if we have geographic data. So there are many different types of modeling questions and challenges in this field. And it is not really feasible to teach single approaches that could approach all of these different aspects at the same time. So this is a real challenge in teaching microbiome data science. What are the topics that we should choose for that teaching, and how they relate to current topical research trends? So we have to have some kind of common overview of all of these, but then at the same time understand the big picture. Often one of the challenges comes from the fact that we have many different combinations of even standard methods. So even if we have good understanding of the methods that we apply, we still need to combine them into workflows. And we can easily obtain huge numbers of different combinations of methods. Some of them will maybe lead to significant findings and others not. So we are arriving at the questions of reproducibility and replicability of the studies. And of course, this is also a general issue in research, but I think this is also a very important topic to address in training. And here is just one example about relatively different results that we can obtain for a given population cohort data from Finland by using two different ordination methods. It's the same data as projection methods, two different approaches. So it can make a difference what kind of methods we use. So how to teach for the students or learners what methods to choose. So microbiome research is data intensive and requires many sophisticated computational techniques and different backgrounds from people who learn these. And we have found out that the workflow method of teaching seems to be very successful often in teaching microbiome studies, because when we teach workflows where students can start from the raw data, importing and processing the raw data, to actually analyzing and reporting it, and can really see the whole pipeline, and then start gradually changing things and improving things and experimenting with different methods, they can see the whole picture at the same time while working on understanding individual parts of the workflow. So I would say that this workflow approach is something we like to promote very much. It is also teaching many useful aspects of such research, like transparency of the analysis, collaborative development of these workflows, and other aspects. We are, of course, not the only ones to promote this. There are other researchers, for instance, colleague Susan Holmes has been writing about similar workflow approaches. We have been teaching these techniques and approaches in various training events. In the latest one was this Machine Learning for Microbiome training school in September, October. There will be some more talk on October 14 in this symposium. So if you want to have a further look at some of these resources that we are building, here is an online page that provides more information and links to the sources. And then my time is over, and I wanted to just have 3 take-home messages. So because there is so many different approaches, we must focus on concepts rather than tools. The tools are changing all the time, but the concepts stay. And then I would recommend to use real workflows with real data, open data, to go through the whole process so that the students can see the big picture. And then embracing open science is a way to improve the efficiency of the teaching and also to teach good research practices. Thank you very much.